Today we are going to learn about DNA transcription in prokaryotes through an analogy of an inflatable water obstacle course. Before we start, transcription is the process through which the information in DNA is copied into a new strand, a strand of RNA. Prokaryotic transcription involves three regions of DNA: the promoter region, which we can think of as the entrance to the obstacle course, the RNA encoding region, which is the bounce castle, and the termination region, which is the end of the course. Rna Polymerase is the main enzyme utilized in prokaryotic transcription, and it utilizes phosphodiester bonds joining RNA nucleotide triphosphates, commonly known as RNTPs. We see RNA polymerase in two forms during transcription: as a hollow enzyme and as the core enzyme. We can think of the core enzyme as a person who is going through the obstacle course. The hollow enzyme includes a sigma subunit represented by the pool float, while the core enzyme is just the person. We can think of the sigma subunit or float as giving the boost needed to proceed. We begin transcription with template binding at the promoter region or beginning of the course, as this recruits us to enter and serves as a site for RNA polymerase to land and begin. The hollow enzyme recognizes the consensus sequence, which is the entrance to the obstacle course, and a sequence of DNA found in segments. In prokaryotes, the two sequences found are the -35 control region and the -10 sequence, which is also known as the tau box. These numbers are the number of bases upstream from the start site of the sequence. These are in reference to the +1 site, which is the transcription start site. The hollow enzyme, specifically the sigma factor, detects these sequences and allows it to drop the core enzyme, bind, and begin. Think of this as the person dropping the float as they enter the course. If the person still had the float, she wouldn't be able to fit through the entrance. A detachment of the float, or sigma subunit, allows elongation to begin. Now that the person core enzyme has entered the obstacle course with the help of the sigma factor, the next step is elongation. We can think of this as the race through the obstacle course in the castle part. It is during this step that the core enzyme races down the coding strand and synthesizes RNA, making an RNA strand. At the end of elongation, we reach the terminator region, which starts termination, the last step of transcription and bringing our fun to the end. As we begin termination, we synthesize an invert repeat sequence as conveyed by the last obstacles. The invert repeat sequence and the obstacles cause a hairpin loop to form in the RNA, which is conveyed by the end tunnel through which we stumble out of. Following the hairpin loop and tunnel, there is a short region of adenosine nucleotides, which we call the poly A tail. As we proceed, we form uracil adenosine nucleotide bonds that easily break apart. These bonds breaking allows for us to leave the obstacle course or the RNA to dissociate from the DNA ending transcription. Through this analogy, I have demonstrated row-independent termination or intrinsic termination. I would like to note that there is another form of termination in prokaryotics, which is known as row-dependent termination, and requires a row molecule to attach to the RNA, chasing after the polymerase, demonstrating Helicase activity and pulling apart the complex at the end, ending transcription. I am not going to visually show this, but think of it as if you were being chased through the obstacle or course and the person caught up to you, ending your fun. Since we had so much fun, we do want to go through the obstacle course again. Unfortunately, a child has thrown up at the beginning, adding 5 base pairs to the promoter region. This would be considered an insertion mutation, and more specifically a frame shift mutation, as this causes all of the following base pairs to be shifted. The prob now tau box would now be at -5, but more importantly, this causes all of the codons and amino acids coded for after the insertion to be changed. This results in a disastrous effect on the sequence and likely results in premature termination due to the missense. Here we see the course being closed and we are unable to continue going through.